I'm going to scare everyone.
Listen, we're recording this right by Halloween. It needs to be scary. So, yeah.
Yeah, well, finding two positions doesn't get more scary than that.
Yeah, that ups the scare factor, right?
Yeah,
Automatic
yeah.
horror show, right. Welcome to Absolute Genius, a podcast series from Thermo Fisher Scientific. I'm Cassie McCreary.
And I'm Jordan Ruggieri. And folks, the time has come. We are back for season 2 of our show. And we have got a fantastic slate of geniuses lined up to join you in your earbuds over the next few months. And to kick things off today, we have not one, but two geniuses with us. Dr. Pavla Brakova and Dr. Nehemiah Alvarez.
Pavla and Nehemiah study reproduction at Eastern Virginia Medical School. Their work focuses on RNA transcription and decay in early embryonic development and the implications this process has for fertility. And they're also married. We had a fascinating conversation with them about their research and their shared life in science, and we're super excited to share it with you.
Talk about a plot twist when Cassie and I realized that they were married and studying reproduction and oocyte decay.
Just a show about geniuses hosted by a couple of geniuses.
All right. Pavla and Nehemiah, thank you so much for joining the Absolute Genius podcast. We are very excited to have you on today. To start off, can you tell us a little bit about yourself and your research?
My name is Pavla Brakova, and I am currently an assistant professor at Eastern Virginia Medical School in the Physiological Sciences Department. I went to college at a very small liberal arts college in Iowa. From there, I went to graduate school at the University of Iowa in molecular biology. I was studying ovarian cancer there. I always wanted to study reproduction, and I didn't get that opportunity until my postdoc at the University of Kansas Medical School. I was determined to be there for 1 year and find a job and start my own scientific career, but I ended up being there for 7 years. And then we were able to find two positions here at EVMS, and so that's my route that I took to this point.
My name is Nehemiah Alvarez. I'm an assistant professor in physiological sciences at Eastern Virginia Medical School. I'm also director of our advanced sequencing program here. I came from the University of Kansas Medical Center, so I did a postdoc there. I also did my graduate school there. Pavla's postdoc took an extremely long amount of time, but my graduate school took a long time. So I spent 4 years in a PhD in one laboratory, and that laboratory subsequently closed down. And then so I had to restart my entire PhD all over again.
Oh,
So initially I was working in RNA biology and Drosophila, and then I had to switch into DNA damage repair in mice. So it was up the evolutionary ladder, but a step backwards in a transcription. I went from post-transcriptional regulation to pre-transcriptional regulation. And so I finished my PhD, short postdoc for 2 years. Then I was on research track faculty for a year and a half, and then we ended up both of us ended up getting a position here.
Seems like a wild path for sure. Can you tell us a little bit about the research that you're doing now and some of the projects that are happening in your lab?
We are interested in female fertility and understanding what makes a good egg. In IVF clinics, when they do in vitro fertilization, the rates of success are fairly low, and even in unassisted fertility, when people try to conceive, the rates of success are also pretty low. And so it's very poorly understood what are the requirements within the egg that will allow it to progress into an embryo and then make a successful pregnancy and make a human child. So that's kind of the big picture.
So a lot of what we work on now is trying to understand how in the egg or the oocyte, what are the mechanisms that regulate RNA stability. And so the reason why we're interested in RNA stability is because the oocytes in the egg are a very unique in cell type within mammals, and that is where there's no transcription. So when we think about a normal cell, it's making RNA, and it's making RNA about the exact same rate it's decaying the RNA. So there's this equilibrium that is occurring. But in the oocyte and then during subsequent early embryonic development, there is no transcription. And when it's going through early embryonic development, so you're with the cleavages, it's still not making any new RNA. And so the cell has to make do with about 250 picograms of RNA over the span of it's like 96 hours without any transcription. Almost like 80 to 90% of the RNA is degraded before the embryo genome turns on to replace that RNA. And what we are focused on is understanding how RNA can be differentially modified and how that leads to that proper degradation. And so the projects that we work in the lab are specifically centered around that. And then one of our other projects is we develop, we work on trying to understand the RNA modifications that in relation to like how we can design effective RNA therapies to treat infertility disorders in the context of the oocyte.
Very interesting. So talking again about that RNA, I mean the RNA is translated into some type of protein and has some type of structural function. Are there particular targets or particular pathways that are of critical interest? Are you looking more at just general quantity of RNA?
In terms of the RNA modifications, what we've seen is that there are global patterns that happen, but not specific targets that are like super important, critical. It seems to be more of a global pattern and
like a mass action. Like you need like general process. Like we look at like how these RNA modifications could impact. We were talking about stability, but they also impact translation. In oocytes, there's a phenomenon that's like translationally coupled RNA degradation. That means for every RNA, when RNA is translated, it is degraded. It's like think about like we made the protein we need. Now we're going to get rid of that RNA so that we don't keep making more of it at the wrong time. The RNA modifications can potentially impact how that RNA is translated, if it is translated at all, and it's subsequently how it's degraded. That's sort of like we know. So we look at these, modification status, and then we look at how that like feeds into this, degradation pathway. And there are other degradation pathways within the oocyte, but the main one that we may focus on is the translationally coupled side, because it's the tools that we have to study that are better developed than the tools to study like degradation pathways that only affect like non-coding.
Can you give us an overview of maybe what a common workflow looks like in your lab and some of the techniques that you're utilizing? What does that workflow look like as you go from egg to data?
For us, we work with single oocytes, and we primarily right now use the mouse model. And so like a typical experiment, you have to hormonally stimulate mice to get them to be at the proper egg stage. So we collect, they're called GV oocytes. And then in those, they have the full amount of RNA before the degradation has started. So we compare those to ovulated eggs after another round of hormone stimulation. And so we can collect single oocytes or single eggs and compare, like for example, we use the digital PCR or RNA sequencing approaches to then quantify the amount of RNA.
Yeah, so that's like the one general workflow we use for like our abundance, so we want to know like,
right.
We were interested in understanding like the half-life of an RNA, like how does its decay properties change over, what does its decay curve look like? In order to measure rate, we need to be we need to use very like tools that allow us to do like be very as quantitative as possible. Paula mentions that we do use digital PCR. Sequencing approaches work, but there are some issues associated with those. That's more of like always a relative abundance. Even the use of UMI approaches within oocytes, just the nature of how they're transcriptionally quiescent, leads to UMI. The UMI approach is not a true representation of the molecules there. It just always means like relative.
Can you define UMI?
Oh, yeah. So that's like a unique molecular identifier tag. So these are just random sequences that you can attach to through library preparations for RNA sequencing. You can attach them to they can be attached at the reverse transcription stage or after you get the reverse transcription stage. And then you attach these, so you're counting what are potentially unique molecules. The UMI counts actually go up. You could think of it as if your sequencing real estate was 1 million molecules and you started with 1 million molecules, you're going to, your sampling of that space is going to be, you're going to have 1 million molecules of sequencing and you can only get 1 million molecules output. You're going to get a distribution of those, not a random distribution, but you get a distribution that represents that input. So that's why we use the digital PCR approaches, because when we do digital PCR, we can actually just count the molecules that are present.
And that's incredibly important in our RNA therapeutics studies because, like Nehemiah alluded to this, but we want to try to replicate the physiological condition as much as possible. And so we are going to be injecting RNA into an oocyte, and we need to know exactly how many copies of that to inject.
Yeah.
And then be able to measure how the RNA modification is impacting the stability of that RNA, the translation, and then when it does reach maturity, make sure that it is undergoing the same decay that is happening physiologically. So we always want to compare to try to not to try to overload the system, because we never know. Even if you can rescue a fertility defect, you might be altering a lot of RNA binding protein networks within the cells that could have other impacts that you don't even measure at the time.
Makes sense. I have a million questions from all of that information. Working with single cells and with RNA can be really complicated, especially when you're looking at degradation, because you blink at it 2 instead of 1 time or 3 instead of 2 times, and the RNA is all of a sudden degraded. Is that something that you run across fairly frequently?
Well, so we did a lot of experiments when we were postdocs looking at the stability of RNA under different conditions, different incubation times, and it didn't seem to be as impacted by temperature, for example. We saw that pH was a big issue, but we did a lot of these types of very basic experiments where we looked at RNA quality after certain conditions, and I think that made us feel pretty comfortable handling it. I think what helps us in the lab is that we got digital pipettes. So they're extremely precise, and from person to person to person, there's very little variability. And so I think our students also feel really comfortable working with it. But there are times when I am pipetting one cell into a tube and it doesn't get in there. I'm not as worried about the RNA as I am about the single cell. We've seen that sometimes there is no cell.
Yeah.
That's crazy. That's just that's awesome as well. Just really, really cool to think about. Talking a little bit about the RNA therapeutic side of it, what are some of the hurdles when it comes to RNA therapeutics for your particular research, and how are you overcoming those?
Just in general, the challenges that would be for the RNA therapeutic in terms of women's health and infertility is that the reason why someone would be infertile is really unclear. That's what makes it the biggest challenge for designing effective RNA therapies. And that's why initially our primary focus is what is the general features of RNA need to be in order for it to be processed correctly by the oocyte. So I think that the biggest challenge is figuring out, what would lead to, a situation in which we would need to use RNA therapeutics in the IVF.
From the literature, it seems to be about 1 to 3% of women in the IVF clinics have a genetic component to the oocyte not being able to mature properly. We're just looking at one very small window of the fertility process, and the amount of women impacted is very low.
In actuality, like if we had, the budget of like $10 million, we could measure the abundance of every RNA in the oocyte. And then we could go back and say, okay, we're just going to make every RNA. We'll just suck out the entire contents of this oocyte and put back all the RNA, and then just
total RNA replacement.
Total RNA replacement. No, and we joke because that even it is like technically it's feasible. Like I mean, it is,
theoretically,
but budgetarily it's not. The ideal situation would be like if we just kind of keep working through this process, trying to figure out at the molecular level, how RNAs are processed. We might just settle on that. Really, to drive these to get these oocytes kind of over this hurdle of maturation defects, you might just only need, a few genes to do that.
Yeah, that's awesome. I want to follow up on the digital PCR elements that you had discussed before. Getting that absolute quantification is really important. Are there any other factors that you consider when utilizing digital PCR, or any other benefits you get utilizing that technology?
In the past, I've done a lot of qPCR in graduate school and in postdoc, and it's always the results and the interpretation of those results, like what does it actually mean, depends on which gene you've chosen, which gene other literature has, like other papers have used to use as your reference, to make it relative to that gene. But it's really unknown how that reference gene is changing over time, how your experimental conditions are impacted. And so it makes it more murky, and it complicates the interpretation and then just the overall entire experiment. So that's another reason why it makes it so much more powerful just to quantify the absolute amount.
Just by the nature of what we look at, we use a lot of single cell sequencing technology approaches. And so this is either through actually the sequencing of the direct, of the oocyte itself, or doing, some sort of, using a single cell sequencing platform. So we can look at potentially what those, like within the whole tissue, what those oocytes are looking like transcriptionally within the tissue, not after we have like manually taken them out and, and manipulated them in some way. Within the ovary, look at, single cells within that tissue, and we look at the oocytes. And then we also look at these oocytes in isolation using just a sequencing platform. And this just gives us a nice large snapshot of what that transcriptional environment looks like, and gives us the ability to look at targets that then we can interrogate in much more precise detail using the, digital PCR. And so we could use it for actually to find targets like that we can go after. The nanopore approaches requires a huge amount of material, and then the digital PCR approaches just need one cell. And so all these kind of work together so we can get, figure out, within oocytes, what's the modification, what's the spectrum modifications across any given RNA. And then we can use the digital PCR approaches to look at changes in absolute amounts of like the enzymes that are potentially responsible for this. So they can try and draw correlations like back through the across our data, just to see like where where could the defect, if we see changes in RNA modifications in our nanopore data, is that correlate with changes in expressions we see across our digital PCR data and our sequencing data.
We hope you're enjoying this episode of Absolute Genius. We are huge fans of digital PCR here and want to tell you about Applied Biosystems' Quant Studio Absolute Q dPCR system. This instrument enables quantification of your targets without the need for standard curves, with excellent precision in only 90 minutes.
You can say that again, Cassie.
Okay, I will. Get dPCR data in only 90 minutes.
Unlike other digital PCR systems, the Absolute Q dPCR instrument does not use emulsion or other droplet-based methods to compartmentalize reactions. Rather, using distribution channels on a microfluidic array plate, the reagent is consistently delivered to physical microchambers. The technology utilizes more than 20,000 microchambers with a coefficient of variation of less than 1%.
Another feature we're excited about is that the Absolute Q dPCR system analyzes more than 95% of the loaded sample, ensuring highly accurate digital PCR results. No more wasting upwards of 60% of your precious samples when loading, which is especially critical for rare targets.
Yes, and Thermo Fisher Scientific has developed a portfolio of robust Absolute Q digital PCR assays. So if you need precise quantification of gene targets for research applications like AAV viral titer for cell and gene therapy research, eDNA analysis, SARS-CoV-2 wastewater surveillance, or liquid biopsy mutation and monitoring, then you'll want to look into this for sure. You can learn more at www.thermofisher.com/absoluteq or visit the Absolute Geneius webpage. Again, that's www.thermofisher.com/absoluteq or visit the Absolute Geneius webpage. The Applied Biosystems Quant Studio Absolute Q dPCR system is for research use only, not for use in diagnostic procedures.
Now, back to our episode. Woo! Welcome to Cassie's Career Corner, the part of the episode where we all don't have to think so hard.
I don't know about that.
At least emotionally, it might be harder.
This is the part of the episode where I don't have to think so hard.
Yeah. Okay. Yeah.
And to our editor, jazzy intro music here. So I wanted to kind of, and I know we touched on this a little bit at the beginning of the episode and everything, but I kind of wanted to go back to your educational paths and your studies and everything. And I just kind of wanted to get to the root of it, of why you've chosen to kind of go not only just down the PhD route, but study what you have, and then, in turn, how you got to focusing in on reproduction and all of this type of thing. Like, why is it that you're so passionate about all of this? I know, Pavla, you mentioned in particular, then also Nehemiah, you as well. Like, how did it all happen, I guess?
For me, it started actually when I was 8 years old. I clearly remember having this epiphany that if animals don't reproduce, that's just the end. Reproduction is one of the most important things. And so ever since I was 8, I wanted to study reproduction. And I always loved animals and tried to find out as much as possible. There was really no other option for me my whole life.
That's awesome, though. How about you, Nehemiah? How did you end up down this path, really?
I just in general just always had a general curiosity of how things work. I distinctly remember this. I was 5 years old. I used to live on a cotton farm, and so in southern Oklahoma, and I was just pouring just chemicals. I'm pretty sure it's like antifreeze and diesel, giving off all these fumes. And I was just thinking, I just really want to know what's happening here. This is crazy. I just always how do things work like that? That just sort of kept, building, as my education, increased, and as I, exposed to more and more different things, that always stuck in the back of my mind. Is, how do things work? In reproduction is an extremely interesting field. You start from one cell and you end up with organisms that are billions or trillions of cells. You could argue that it's pretty niche. Everyone, it's a very tight-knit community.
Yeah.
Everyone probably could trace their entire career trajectory back to maybe 5 people. It's actually a very small. It's a very small field, and so you always, but it's very exciting. I think it's very fun.
First of all, it is my personal quest on this show to help solidify the concept that science is the coolest. So thank you for contributing to that. I said on our very first episode, I called science the punk rock of academic disciplines. Its backbone is curiosity. It is the epitome of screw around and find out. Science is the coolest. So we've reinforced that. Now, what you're saying about this being a tight-knit community, something that has come up time and time again in my career corner discussions is kind of the importance of mentors, but also networking and that type of thing. And being in the field that you're in, having it as kind of a relatively smaller bubble. What is finding a mentor like? How do you hang on to that mentor? And then on the flip side of things, I would imagine now being in the positions that you're in, you're starting, you're mentoring people as well. What is all of that like?
In general, it's really important to find a mentor who is supportive of your career choice. And I think so I think that in my observation from graduate school through postdoc and even, early, even my junior faculty right now, is that most trainees in a position, they'll always say, like, they'll be very candid. They'll say, I don't know what I want to do. Like, that's an honest thing. But I think that what really helps, like where I see people who are very successful, is people who internalize that thought and instead just say, this is what I want to do. You're 100% committed, and then it's okay to change your mind, but it's like, make sure, like, it's just like you know that your mentor will support you in changing your mind.
Yeah, and like how Nima was saying that you just have to go 100% in if you know that that's what you want. We were told, don't study the same thing, don't be in the same department, don't be in the same lab.
Take that.
Like,
And
we had a vision, and that's the only thing that we wanted to do, and so we just tried as hard as we could to make that happen. And even though we are here, it almost didn't happen. Go after your dreams. I mean, it's corny, but you have to do it. If you know that you want to do it, you have to do it.
Yeah, that's sage advice. I think we only have about 10 minutes left, so there's one more question I want to just make sure we fit in, because it's my favorite question, so selfishly I want to ask it. And I ask everybody, what is your best or favorite or proudest lab moment? And what is your biggest lab mistake, oops, screw up, oopsie daisy, whatever you want to call it?
There's two things I remember as my biggest oops. One that's most vivid in my mind is the most recent one, in which I was doing an RNA sequencing library preparation. And like we talked about how important it is to have like an internal normalizer, so in oocytes during the RNA decay process, so that you can quantify a little bit more accurately how many transcripts there are of a certain isoform, and that's called an ERCC spike-in. And so it's a standard RNA that you can buy from a company, and you have to spike that in at the beginning of your experiment so that it undergoes the reverse transcription, the amplifications, everything, just like all the other RNA, and you can compare to it. I forgot to put that in there. So that was a whole experiment.
All right.
Yeah, I did also mess up the buffers, which Nehemiah had to spend like an hour conversation with the sales rep or the scientist at the company, and Nehemiah was able to fix it. And then I also remember in graduate school I was loading a 384 well plate for qPCR, and at the very end somehow it slipped and the whole thing just splashed into my face.
Oh,
so that was a day I sat there and I cried, and I just went home.
That's sad. All right. And on a positive note then, what's your proudest? What's your happiest?
When I'm happiest and kind of most excited about the research is when I'm writing grants and talking about the ideas and developing the projects. That's what makes me the most excited. And that's kind of how we divvy up a lot of the work, because we run a joint lab. And so we have independent offices, independent space, independent funding and everything, but we do everything together. We submit everything together, and we are a joint unit in like every way possible. A lot of the way that keeps our lab moving forward, especially now beginning a lab, is a lot of the writing. I'll spend the time writing, and then Nehemiah will be doing the experiments. And so that's kind of how we divvy up the work. That's what is kind of the most enjoyable process to me, is developing the ideas and talking with Nehemiah about where we're going to go with our next project.
All right. And Nehemiah, when have you screwed up?
I'd been right when I started grad school, or maybe when I was a technician. I was doing a lot of, I was in a laboratory. We're in RNA biology, so we always had to like make our own antigens and make our own antibodies, purify them, and do a lot of protein, like, not like hardcore biochemistry, but do a lot of protein biochemistry. And so a lot of this involved a lot of cloning and molecular biology. And I remember the, going through all these steps of, cloning the gene that we were interested in, and going through the whole process of cloning it into a vector, and going through the whole thing of like expressing it in E. coli and purifying it, and with this whole thing. And and just nothing was working. Right, we couldn't get we couldn't see the protein on some protein gel. Nothing was expressing or changing like the temperature, doing all these kind of weird things. And I mean, just like months of like troubleshooting. You go back and look, and I forgot to add the Cossack sequence to the. So this is an important ribosome entry sequence that tells in eukaryotic organisms to start translation, and all of this was precipitated because of a lack of a control, right? Like I didn't load a control on a gel initially, and so that would have told us immediately what was wrong. Instead, I was operating under the assumption that it was expressing, and we were just doing something, it was expressing in the E. coli, but it was just precipitating. It wasn't soluble. So we were doing all these, crazy purification strategies and dialysis. And I mean, it was just, colossal waste of time.
How about proudest or best?
I think honestly, just getting us in a joint, like a joint position. I think that was like one of the most, it's a relieving, but also exciting, because then we can continue to do research together. It would have been a very different experience than it is for us now. I mean, the only time we actually end up really talking about science is actually at work. Our kids are we have to, focus on them. So
they're like, no,
stop talking about that. Stop talking about it. So we're like, okay, fine.
Pavla and Nehemiah, thank you so much for joining us for Absolute Genius. We really enjoyed having you on.
Thanks so much. We also really enjoyed our time with you.
That was Dr. Pavla Brakova and Dr. Nehemiah Alvarez at Eastern Virginia Medical School. Thank you so much for joining us for today's episode of Absolute Genius. We have much more to come this season. This episode was produced by Sarah Briganti, Matt Ferris, and Matthew Stock. Stay curious, and we'll see you next time.
No, this is the same. I just wear a blue shirt, so.
Well, Nehemiah has the Obama strategy where you have the same
colored shirt, if I just changed my top.
So you're like a cartoon character. You open up your closet, that's just like 40 of the same. Yeah,
you open his closet, and it's like the same exact outfit.
I just changed the tie. That way you know what day of the week it is, because the tie will just change.
There you go.